Well, welcome to Foresight Neurotech Group. I'm really happy to have really the first one of our 2024 seminar series. Really delighted to have Anthony Zador. Thank you so much for joining us. You'll be discussing brain wiring through the genomic bottleneck. I'll be sharing a lot more info about you here in the chat. I'll be sharing more info about future seminars upcoming in this series in the chat because we already scheduled into Q3 at this point. And yeah, thanks everyone for joining again. Thank you so much, Anthony, for joining here to present. And if you have any questions, feel free to already drop them in the chat. I will surface them to Anthony in the Q&A. And you can also raise hand if you want to ask the question directly. Thanks everyone for joining. Thank you, Anthony. And the stage is yours.
Great. Thank you. Let's see. Normally, if I'm giving a real talk, hold on, I'm just trying to position the window here. Can you guys all see what I'm?
You have to share. Yes.
Arranging my windows here. Normally, I invite interruption, and I'm happy to have interruption questions during the talk. This makes it a lot more fun if it's interactive. I don't really know what your etiquette here is, but do feel free to interrupt with questions. I'll be trying to look at the chat as well, though that's a little bit harder. Anyway, so my name's Tony Zador, and I'm going to be talking about some technologies that my lab has been developing for the last 10 years, a little more than that now. And our basic goal is to develop technologies for understanding brain wiring by converting the problem of brain wiring into a problem of high throughput DNA sequencing. And I'm going to also be talking about basically why we developed this technology, given that my background was originally in machine learning and then in neurophysiology. Okay. So this actually is a summary of the talk. Basically, what we like to do is take brains, endow individual neurons with specific DNA barcodes, grind up the brain in various ways, and then reconstruct the circuit from those barcoded neurons. But I'll provide a little bit more meat to that in the coming few slides. But So I'm going to start just by pointing out something that we as neuroscientists often take for granted, but is less obvious for communities that are further away from neuroscience, in particular for machine learning communities. And that is that a great deal of animal behavior, and I dare say human behavior, is innate. So here's some examples of that. So termites build mounds. That this thing in the upper left is a big termite mound. It's innate in the sense that if you start from a single termite egg for the queen, she'll generate a whole nest of termites that will eventually build a mound like this. In the bottom left, animals, some kind of a goat-like species competing in mating season. Spiders hunt and know how to build webs and know how to hunt immediately after birth without instruction, without learning. A colt can walk at birth. What all this goes to say is that although in machine learning, as the field itself is called learning, is based on the idea of learning, nonetheless, learning in some sense is overrated. And a lot of what we do, what all animals do, is innate, encoded in the genome. And here's some great examples of that I really enjoy looking at. If you have a chance, I recommend looking up a video of Justin Beaver, the rescue beaver. And he was rescued soon after birth and adopted into a home. And he fairly quickly adopted the behaviors that are natural to its species. That involved, as you see on the right, trying to gnaw down the wood that builds the house to build the dam. He did find a lacrosse stick, and eventually he took all these things and tried to put them in the bathtub to build a dam. That was not learned from his parents. There were no other beavers around. This has been studied in detail in the lab of Hopi Hoekstra at Harvard, who studies two different species of mouse, Paramuscus, one of which builds a long tunnel, an escape tunnel like the one on the left, and the other of which builds a short little stubby tunnel. And what's interesting is that these two species are close enough that they can not only interbreed, but they can be cross-fosters. And the key fact is that if you take the mouse from one species and have it raised by foster parents from the other species, they will build the tunnel that their biological parents built, not the ones of their foster parents. So somehow built in the genome is knowledge of, or somehow the motivation or desire to build the one type of tunnel rather than the other. So where do these behaviors come from? They come from our DNA. So we all start from a single fertilized egg, which contains the DNA from both parents. And somehow from that egg, the entire body and eventually the entire brain with all its circuitry is encoded. You can ask a question, how complicated is that brain generating program? And you can actually get an estimate of that by quantifying it. And one way to think about the complexity of the final brain is what in neural networks we'd call a weight matrix, which can be represented as a list of connections among the neurons in the nervous system. And you can do a little bit of math and conclude that the complexity of a random matrix when expressed in bits is proportional basically to the size of the weight matrix. You can get a little bit more precise on that, get a somewhat better bound if you have a sparse matrix, which is the kind of matrices that you find in the nervous system, because most neurons make only a small subset of possible connections. Most of the information that it takes to wire up a biological brain goes into specifying which are the partners, which are the subset of neurons out of the entire brain that a given neuron is going to connect to. And so you get this formula here, which is that the complexity in bits is approximately equal to the number of synapses times the log of the number of neurons. So let's apply that to a couple of examples. C. elegans, the well-studied species with 302 neurons and 7,000 synapses. And it has about, if you run through that equation, you see that it takes about 60,000 bits to wire up its brain. So is 60,000 bits a lot or a little? The C. elegans genome has about 100 million bits. So there's really no problem there. Every connection in C. elegans could, in principle, be explicitly specified in the genome. Now, that's not exactly what happens, although it is the case that the wiring diagram of C. elegans tends to be highly stereotyped. But there's really no mismatch here between the size of the genome and the size the complexity of the connectome. We're in a very different situation when it comes to mammals, and in particular, the human brain. So the human genome is somewhat larger. It has about 10 to the 9 bits, but the human connectome is far more complex, where if we take that estimate from before, it would take about 10 to the 15 bits to list all the connections in a human brain. So there's this tremendous mismatch between the number of bits available in the genome and the number of bits it would take to completely specify a brain connection by connection. So what is going on there? There seems to be some kind of what we call a genomic bottleneck that takes a much smaller genome to the much larger connectome. Now, is there really a mystery there? In large strokes, we know that we know the answer. There's no fundamental mystery. The answer is that what's specified in the genome is not simply a list of connections, but rather a set of rules for wiring up the brain. And we can illustrate that by thinking about the difference between a random network, such as the one on the left, and the highly structured network shown on the right. So the one on the left, you can see that each neuron, just like the one on the right, has 4 connections, but in this particular case, the 4 partners are assigned randomly. So you can see each one of these neurons has 4 lines coming out, but they're chosen randomly out of all the 36 possible, 35 possible partners. Here on the right, it's highly structured. So on the left, in order to reproduce that particular network, you would have to actually list all the connections in that matrix. Whereas the one on the right, you could specify with far more ease simply by saying, connect to your 4 nearest neighbors. So that is one of the main considerations that got us thinking about what are the rules that are used to wire up a cortex. And basically, just to put this in a larger context, the way we think about it is, I guess I'm just summarizing here, that we have a genome and that the genome contains a set of rules rather than a list. I'm going to describe now some of the technologies that my lab has been developing for actually converting this problem of brain wiring to a problem of DNA sequencing. So what is challenging about the problem of measuring circuitry? It's illustrated here. This is a picture of a bunch of neurons in the cortex. Each one of these is a neuron. And the problem, of course, is that the axons are all tangled together. We'd like to be able to trace each one of these axons to all possible partners, but the only technology available right now for doing that completely and perfectly is electron microscopy, which, although it's been improving, remains slow and expensive and requires very expensive equipment. The inspiration for our approach actually came from the work of Jean Livet, Jeff Baines, and Jeff Lichtman. And it was a paper they published 15 years ago now called Brainbow. And there they used a very clever genetic trick to label each neuron with a unique color. And the way they got the unique color is actually through a combination they expressed a combination of genetically encoded fluorophores. And they, through clever genetic recombination tricks, they were able to develop a palette of about 200 different colors. So that was brilliant. And it really impressed me, especially because the technology back 15 years ago for doing that was absolutely cutting edge. The problem with it, the reason that, although it makes beautiful pictures, it has and has had applications, it really hasn't completely taken off, is that you can't really easily resolve, in fact, you can't, not just easily, you can't resolve 200 colors. So even though in principle, there are 200 colors expressed in practice using microscopy, you can only resolve maybe 3, 4, or 5, or maybe possibly 6 colors. And the reason is that the fluorophores that are expressed have very wide spectra. And so you just can't distinguish different combinations beyond a few. So our solution to that is to replace colors with numbers, digital Brainbow. And instead of using colors, use arbitrary DNA sequences. So the idea is to have each neuron express a unique sequence of DNA, genetic barcode. Now, barcoding of various sorts these days has become increasingly common, but at the time that we undertook this, it was actually pretty challenging to think about how we were going to do that. And I'm going to describe some of those challenges. So the advantage, of course, is that the theoretical diversity here is basically infinite because, yeah, here we go. Because the diversity scales exponentially with the length of the sequence. With one nucleotide, you get a potential diversity of 4 because there are 4 nucleotides, ATGC. With 2, it's 4 squared. And by the time you get up to 10, you get a potential diversity of 10 to the 6. And by the time you get to 30, which is how many we have in our barcode, we have a diversity of 10 to the 18. So if you were to label, let's say, 10 to the 7 neurons, which is about how many there are in the cortex, if you had a diversity of 10 to the 9 barcodes, that would be sufficient to label them, all the neurons, with very little chance that 2 neurons would randomly have the same barcode. So that was the core idea, but there were some technological challenges ahead of us, and those were really addressed by a student, Ian Pycon, for his PhD thesis. And so the core question that we had to address was, how do we get the barcodes from the stoma, which is where we're going to express them, out to the axons and dendrite, which is where we needed them. And the idea that Ian had was that he would express the barcodes as RNA and then drag the barcodes out to the axon terminals. And so what he did was he engineered a protein, a modified synaptic protein, and that's shown here on the right. And that protein had an RNA binding domain, and the RNA that we engineered had we put a specific hairpin called a box B that bound tightly to the so-called N lambda domain that we put on the protein. And so we now have a system from this, where we have proteins that traffic out to the synaptic terminals, that traffic out to the synaptic terminals and drag the RNA barcodes out with them. So that also leaves the question of how we get these barcodes into the neuron. The way we chose to do it is we engineered a pool of viruses where each virus expresses a unique barcode. Each little virion expresses its own little unique barcode. And the reason that's relatively straightforward to do is that we can generate viruses in the lab from a plasmid. So that converts the problem of barcoding viruses into a problem of barcoding plasmids, which is pretty straightforward. So basically, we have a pool of plasmids, we barcode them, then we convert the plasmids into viruses, we make the viruses, we inject them into the brain, wait 2 days, and then we now have barcoded neurons. I should say that although by far the most common virus that people use in neuroscience these days is AAV, or adeno-associated virus. For these experiments, we used a different virus called SYNVIS, which has the advantage that it expresses at very high levels and is very easy to engineer. And also it doesn't typically require a high multiple of infection, MOI, the way that AAV does. Okay, I'm going to tell you about some of the first experiments that we did at the very first, one of the first applications of this a few years back. And that was in collaboration with Tom Mercer-Klogel's lab. And this is work of a graduate student in the lab, Justus Kempschel, who now has his own lab using some of these techniques at Johns Hopkins. And so the basic approach, so we were interested in this experiment in the projection of neurons in the primary visual cortex of the mouse to neighboring areas. And so this big center patch here is V1, and there are 6 higher order visual areas to which these neurons project. And the question we were interested in were the patterns of projections of individual neurons at high throughput. And we injected a pool of viruses to a particular location in V1. And then what we did was we detected out these higher order visual areas, which go by intuitive names like PM, AL, RL, AL, LM, and LI. And the question is, does a given neuron project to one of these only, to multiple ones? Is it random or what? In order to answer that, we injected the virus, waited 2 days, and dissected these areas out, and then collected the material from each one of these into tubes. And then we looked at what the sequences were in each one of these tubes. So if in a given tube of, let's say, we have barcode 388, we know that neuron 388 projected to RL. Do we see 388 anywhere else? Actually, I don't. Yeah, but here we see that neuron 007, the secret agent neuron, projected to both LI and LM. And these numbers here show the number of times we see a unique copy of that barcode. And that's important because that tells us something about the strength of the connection. Okay. At this point, people usually raise their hands, or at least thinking about all sorts of technical concerns. The first one is multiple barcodes per neuron. And although we try to avoid, it's not actually the worst problem in the world, because if we have multiple barcodes for a given neuron, then typically that just means that we see that neuron twice in these experiments. In later experiments, we actually have a way, when we do spatial transcriptomics, we can actually see those barcodes in the given neuron. But with the bulk sequencing approach that we're using here, even then we just see the same neuron twice. On the other hand, if we have insufficient barcode diversity, then that's terrible, because it means that we will merge the projection patterns of 2 neurons into one. That's why we're very careful to have sufficient barcode diversity, so that the chances that the same barcode will infect 2 different neurons by chance are extremely low. The sensitivity of this approach turns out to be as high or higher than most conventional approaches. And that's simply a reflection of the fact that DNA sequencing is highly sensitive and in fact based on single neuron. Now, one question that often comes up is basically the fibers of passage problem. So we have barcodes that are inside of axons, but we can't ensure that a barcode, just because it's in an area, is actually making a synaptic connection with any of the targets. And that's a true fact, but it's also a fact that's true of many, arguably most sequencing techniques, including conventional GFP tracing, as is found in the Alle Institute. I'm happy to return to these technical concerns and discuss them in more detail during the question. We have a question from Logan. I'm happy to take a question now.
Yeah, hi. I was just wondering about the viruses used, and you mentioned AAV, in which case it makes sense that the plasmid barcoding would work, since you could have a plasmid pool of random plasmids and they're transfected directly to produce the AAV. But I was wondering about viruses like adenovirus that require upscaling, so it's iterative growth stages, and if that introduces bias.
That's a great question. So the answer is that basically we have to be very careful when producing the viral library to not allow the pool to pass through a bottleneck, a barcode bottleneck. So for reasons that we don't yet fully understand, for years we've been trying to get this to work with AAV, and it doesn't work with AAV, regardless of any problems having to do with barcode diversity. The barcode system, when expressed with AAV, simply doesn't drag RNA out to the synaptic terminal. We could talk about my speculation about why that is, but AAV seems to be a non-starter. Where this is a real problem is with rabies virus. And it's actually, we now have several projects. I'm collaborating with several labs that are barcoding rabies virus. And indeed, because of, in fact, I think the severity of that upscaling, as you call it, is very great with rabies. That's been a real challenge. And so basically the answer is go back to the virology, figure out why the efficiency of going from plasmid to virus isn't higher and try to make it higher, or just brute force it and make huge vats and plate them out. So that has been our approach. So we are now, in collaboration with these other labs, beginning to see barcoded rabies. In fact, my former postdoc, Diane, has a paper that I can't remember if it's out yet, but it's on bioRxiv showing some efforts along these lines.
Very interesting. Thank you.
I should say that we have an NIH fund. Oh, do you have another question?
I was just popping in to remind people that if they have any other questions, please feel free to raise hand or put it in the chat so I can ask it. I have a whole spot of questions, but please make your point, Anthony. I didn't want to interrupt you. Sorry.
Sure. Feel free to interrupt me, or we can do it anyway. I'm happy to have a more interactive thing. I don't feel compelled to get to my last slide. I think it's hard. Like even when talking to a real audience where I can see faces, I invite questions. And on Zoom, I'm like talking to the void. So it's always tough. I once actually spoke for an hour and discovered that my connection had died. And it was really it was a sad moment.
But no, we're all here. And we're definitely listening in. So if you want to get through the last,
even if you weren't there.
If you want to get to the last bit of the slide, that'd be wonderful. And then we can properly open it up for discussion.
Okay. Then I'll plow forward. We have a MAPC core facility at Cultivate Harbor. We've used it on lots of brain areas in lots of species, and we're always excited to go into new brain areas and new species. So returning to the thread, you might remember a few slides back, we had just injected barcode into the visual cortex, and you were all in suspense to hear what we found. And so here's what we found. So here's an example of the kinds of data that we get. Each row here is the projection pattern of an individual neuron. In this case, from a single sample, we had 533 neurons, and each neuron projected to its own set of targets. You'll see that, for example, a lot of them projected to LN. This one here, I'm not sure if you can see my cursor, but you can see there's one that projected to LI and LM, and, for example, didn't project to RL. And here we have this is quite the raw data, but this is the result of an experiment. And yeah, here are examples of what these projection patterns mean. The injection site is here. This neuron projected to these 3 areas. This middle neuron projected to these 2, and this top one projected to the 2 in the upper left. So how do we analyze this? There are lots of things you can do with these data, but one question that we found particularly interesting was the extent to which these neurons were projected in a structured way or randomly. So the null hypothesis here is that you would be able to predict the projection pattern of a given neuron by knowing to a pair of targets, by knowing its projection to the average projection of neurons in that area to those 2 targets A and B individually. So for example, if the projection probability of neurons in the source was 0.5 to target A and 0.5 to target B, you'd expect on average that you'd get 1 neuron that projects to both, 1 neuron that that projects to 1, and then 1 neuron that projected to neither. That's the null hypothesis here. Alternatively, you could get that same overall projection probability of 0.5 by in an extreme situation where each neuron projected either to both or to neither, but no neuron projected to just 1. And so we can systematically go through and ask whether projection patterns are over or underrepresented, or whether they can be described by this null hypothesis. And what we found was that certain projection patterns were greatly overrepresented. In fact, my red oval skipped the LM-AL overrepresented projection pattern, but you can see that a significant subset were overrepresented, a handful were underrepresented. And that's summarized here. We find these motifs. And so that led to the working hypothesis in the lab that the projection patterns are structured in this way. Okay, so projections are not independent. So that was the state of affairs circa 2018. We've now moved forward a couple of years. Actually, I guess this is an outdated slide. I thought I had updated it, but I should say 2024. We've moved even forward in the calendar. And so in this experiment, what we did was we mapped all of cortex. This is work of a postdoc, Long Huang. We mapped all of cortex or at least half of cortex in the mouse in a single experiment. And the way he did that was he made 600 independent injections into one hemisphere, waited 2 days for the barcodes to be dragged out, and then cut the cortex, spliced it, and then collected about 300 voxels, or we call them cubelets, and analyzed the projections from each cubelet and every other cubelet, and registered them to the Alle atlas, as you can see here. And that led to a data set here that has about 200,000 individual neurons. And for each neuron, we have its projection pattern to all the other targets that we selected. And you can see here that we selected 2 just as an example, but sitting on the disk, we have the projection patterns of all 200,000. And so out of the many things that we could ask about that, there were a couple of interesting statistics that really stood out for us. One that came as a surprise to me was that a typical cortical cubelet at a resolution of about 1 millimeter, as shown here, projected not to 1 or 2 or even a handful of other targets, but to 70% of all possible targets. I was surprised at the extent to which the cortex, it's much closer to being all to all than I would have thought. On the other hand, how to reconcile that with what you typically see when you inject GFP into a particular brain area. And the way to reconcile that is turns out that although a given cubelet projects to 70%, maybe more, that's just the lower bound of all possible targets, the majority of the projections are to a small handful of targets. 20% of the targets account for 85% of the projection. But returning to the question of I'm going to skip this and get to the key fact, which is, I'm going to turn to the question of motif. And the key fact in terms of motif is that for long-range projection, contrary to our expectation, we could actually explain the all-to-all connectivity with the null model. So this is actually at odds with what we expected from the analysis of the local connections within the visual cortex. And so it turns out that only less than 1% of all connections were overrepresented in terms of brain areas. The vast majority could be explained by the simplest model, which is that if I tell you the projection of kubelet 23, the probability that kubelet 23 projects to kubelet 45 and 46, then the chance that a given neuron projects to both of those is given by the independent probability of projecting to one. So that's, to step back a little bit, what that says is that the rules for wiring the cortex are actually surprisingly simple. All I have to do, at least in describing cortical connection, is tell you the probability that a given neuron projects to areas A and B, and I can tell you the probability that a given neuron projects to both, and that's summarized here. I was going to talk about some neural network modeling we did, but I'm actually just, without going into details about that, I'm gonna just say a few words about what this means in terms of what it takes to wire up a brain. I mentioned before, I motivated this by talking about the genomic bottleneck, and a lot of the work in the lab now has been pursuing this idea of the genomic bottleneck, the idea of compressing a wiring diagram as a regularizer for neural network. And one kind of way of compressing this network is by using a sort of probabilistic wiring diagram where you generate networks, you draw networks from a random distribution, and the rules for that are based on cell type. And we've built artificial agents here, and I don't have time to tell you about this, but I think what's interesting to think about from a biological perspective is that we are usually in biologists or describe the tension between genes and environment in terms of final outcomes. And so here's an example of identical twins who have the same genes, but led very different lives. And you had the woman on the right who was a sunbather and a smoker, has very different skin from the woman on the left who was neither a sunbather nor a smoker. So this is clear evidence for environment in producing outcomes, at least in terms of skin looks like. But there's another example of, this is a fascinating book written by a woman whose identical twin ended up with schizophrenia. And I think one possibility for what leads developmental, identical twins have about 50% concordance with schizophrenia. So if one identical twin has schizophrenia, there's 50% chance the twin will. And one possibility is that differences, since they have the same genes, is not due to environment, but rather to a third factor, which is just throws the dice when the brains were being wired up. And that's illustrated here, that there's a third factor in thinking about the development of neural circuits, which is chance one. So I'm just going to say a couple minutes about some further technology here. So to summarize what I said before, innate behavior is propagated across generations via genomic bottleneck. I guess I'm running out of time here. Is that right? So I'm going to very quickly just say that we've developed a technology for in situ sequencing, which produces a much higher resolution. And it's basically but I want to leave time for questions. So I'm going to very quickly run through this and then go back. So in situ sequencing. So all of what I've talked about so far is MapSeq, which is limited by the crude spatial resolution. We have to do dissection, and also we can't learn much more about the cells other than their barcode. And in particular, we can't learn about their morphology or their cell type as determined by gene expression. And also, I'm a physiologist at heart, and I would like to be able to relate all the things we learn about neurons to their neural activity. We developed a next generation tool called barcoding. And basically, the way that works is by imaging. And what we do is we use Illumina imaging technology except we do it in situ. So basically, here's an example of a bunch of barcoded neurons. And at each cycle, what we do is we figure out what the next nucleotide of the barcode that we're sequencing is. So here are 4 possible colors associated with the 4 nucleotides, A, T, G, and C. Here are 2 neurons, and you see in the first cycle, that neuron's barcode on the upper left is an A, then a G, then a C. And so by successive rounds of imaging, we can actually figure out the barcode expressed by that neuron. And we've now scaled that up considerably and applied it to genes. So here's the paper that my postdoc Xiaoyan led the work on. It's actually just accepted for publication, but it's currently available on bioRxiv. And so we can now go through an entire brain in less than a week for about $2,000 and get over 1 million neurons and 150 genes per neuron. We can also apply this to barcodes. And I'll also say that we've extended this to axonal resolution. This is some work that is done by a postdoc, Li Yuan, in the lab. And you can see that here there are about the projection patterns of 100 neurons out of a few thousand that we labeled. And for each of those neurons, we can see where it projects. So it's basically it allows us to look at the projections of lots of neurons all at once at sort of axonal resolution. And the last slide is that we can combine this with activity and in vivo imaging. And right through that last part, but I just wanted to thank the people who actually did the work here, in particular, Eustace, Ian, this Capsule, Ian Taikan, Longwen Huang, Diane Chen, King Zhang, who runs the facility, and my funders. And sorry for not leaving more time for questions, but I'll stop here.
Awesome. That was wonderful. I'm sure we have plenty of questions to go through now. I tried to find the paper that you just referenced. I hope I posted the correct one. I think so, but if not, then please correct me. Okay, we have a bunch of questions already. Are you okay with us just jumping in?
Yes. So that paper that you posted is the last paper that I showed with the axons. I can show you the one I'll find the one with genes and post that one as well.
That would be great. Okay, good. Okay, super. We have first one up, Nicola, if you want to unmute.
Sure. Awesome talk. Thanks a lot. I have a question, but I just wanted to mention before that, are you familiar with the nanostring COSYNE technology for spatial?
I'm sorry, with the what?
Nanostring, they have a tool. It's called COSMX, and it does spatial transcriptomics. It could be useful.
There are, at this point, at least a half dozen or a dozen different technologies for doing spatial transcriptomics. And it's an incredibly active field. And I think there are advantages and disadvantages to each one of them. We developed ours because none of the existing technologies actually was suitable for sequencing barcodes at high throughput and high resolution. And then later we realized that it was actually generally useful and effective for sequencing, for multiplexing gene sequencing. So I would say that right now, one of the leaders there is Murfish, but that's, I think, in part because it was early to the market and there's a commercial, but we could talk offline about the relative advantages and disadvantages in terms of cost, throughput, sensitivity, error rate, all those questions.
Yeah. So my question is, when you made the observation about the dense connectivity in the brain, that doesn't sound extremely surprising to me because Burkhalter has published that years ago. But I was wondering if you have some observations about bifurcation once you read that onwards.
So like the surprising observation was that for local connection, what we found was that bifurcations were often over or underrepresented, just as we found in area P1. But for long-range projection, the null model was essentially could explain all the data. And so there are a couple possibilities biologically for why that would be the case. One possibility is that it's innate, that for whatever reason, local projections are more finely specified in the genome and that the genome doesn't bother to specify long-range projections at these higher resolution. But I think what's more likely is that the null model of random bifurcation of the null model holds for all projections, but then use-dependent correlations either promote or discourage certain pairs of connections from being formed. And one piece of evidence for that is that one highly overrepresented set of bifurcations are to IPSI and contralateral homotypic targets. And those, you might expect that the activity, in fact, it's known that the activity of homotypic areas are correlated. Whatever the processes are that stabilize connections to one side might tend to stabilize the connection to the other. I guess my model is that the brain starts off with knowing approximately what probability a given area should send to any other target area. And then over the course of development, those are somewhat refined.
Any follow-up? Okay, cool. Next one, we have Logan again.
Yeah, something I've been thinking about that was inspired by your work is the idea of trying to turn connectomics, not just projectomics, but connectomics completely into a sequencing problem, and to still include morphology as part of that. And I've seen some papers, I think DNA-based sequencing or DNA-based imaging, actually. And I was inspired by some of these different areas, and I've been throwing around in my mind this idea of that I think is very technically challenging, but might be cool, of embedding the tissue in a structured DNA gel where you can know where you'd have to be able to reconstruct the gel after it had been disassembled, so that once you sequence it, you can base the reconstruction on, okay, where is this arm overlapping and so on and so forth. But if you could have some kind of a lipid responsive, lipid activated DNA editor, then maybe there'd be some kind of proximity induced activation of this DNA editor to change the DNA or part of the DNA gel at that location. And I'm just wondering if you've ever thought of anything vaguely resembling that, or if you've ever tried anything along those lines, or what.
I haven't. I'm not sure I fully understand what you're proposing. It sounds really interesting and somewhat ambitious. One thing, but I'd be happy to discuss it more. It sounds really interesting. One thing that we have thought about doing is figuring out how to feed little bits of tissue with barcodes and then have the barcodes extend within processes post-mortem. And we made some progress on that, and it's been back-burnered. And I'm happy to tell you offline about some of those ideas. I'd be interested in hearing more about what you're discussing. If you want to drop me an email.
Sure. I definitely do that. Are you on LinkedIn as well?
I think you can, if you Google me, there aren't that many Anthony Zador.
Okay, yeah. I should be.
Zador at CSHL. Here, I'll just put it in the chat for anyone who wants to.
Thank you.
I also dropped a link to your bio earlier in there that also, I think, has your
Yeah, I am on LinkedIn, but I don't really check it very often. So.
Sure. That's fine. I just figured it might be an easier way, but if it's not, then I'll just email.
All right, wonderful. So if anyone else has another question, please feel free to interrupt me, or if you, Anthony, really want to get another point across. Otherwise, I think like a few bits that people are usually interested in faucet salons is just, if you think about your work on the long term, and I know that I guess Logan has already offered a pretty ambitious version. You also alluded to a few implications that it has on the long term, especially I think as we perhaps more about the ML aspects as well, and some of the work that you at least really mentioned with like neural network modeling and artificial agents. Could you make a few more suggestive regulations about where this work could be going in the next 5 years or something?
Yeah, on the experimental side, what we're super excited about is combining I split it to this slide so quickly, I doubt anyone saw what I was doing, but what we're really excited about doing is combining the in situ sequencing, which allows us to figure out the projections and gene expression with in vivo measures of activity. And so the idea is, in fact, we're making progress on this, to be able to record from a bunch of neurons in vivo, let's say in visual cortex, and monitor their response properties, and then be able to say, this particular neuron, which we recorded in vivo and responded when the animal was sticking out its tongue, this is a neuron that projects to the tongue area, right? And it expresses the following gene. And so that combination of that way of getting a richer picture of each cell, we think is what's needed and on high throughput. In principle, currently we're doing hundreds of cells per animal, and we hope to be able to scale it up to thousands of cells per animal, maybe tens of thousands. So basically you get the circuit diagram along with the activity of each neuron. So that's one big direction. And on the theory side, what these high throughput techniques are giving us are constraints on the kinds of wiring rules that are at play either during development or in the adult animal. And that sort of gives us hints about what it takes to build a brain. So the core idea of the genomic bottleneck is that the rules for wiring up a brain are simple, a relatively small number of parameters. And if we can figure out what those parameters are, like not necessarily the values of them, but what the degrees of freedom are, then we can potentially use those as constraints for building artificial neural networks that behave more effectively.
Oh, but I think we have to leave it at that because you said you have a hard stop and we want to leave it at that.
Yeah,
Yes.
Thanks so much. This has
you
been great.
Thank you so much. This was absolutely fantastic. Thanks everyone for your great questions. And I hope it wasn't the last time that we hear from you. Thanks for your wonderful work. Thanks everyone. See you soon.